Perfect. Okay, I think I will start with the introductions while a few more people joined. It is really our pleasure to have Stuart today. And you know him well, but just for a few, maybe are not familiar with his fantastic career. He's a professor and a STEP endowed chair, founding director of the Neurodegeneration New Medicine Center at Scripps Research. He's also a clinical neurologist at UCSD, an adjunct professor of neurology at Yale. And you might know his work very well. He discovered the post-translational redox modification known as S-nitrosylation and have been very, very busy and prolific in that arena. And this is important and is associated with many functions in health and disease. And recently, his group also showed that aberrant nitrosylation can be triggered by environmental toxins, including air pollution. You might be familiar with Stuart's work as a precursor of a patented FDA-approved AD drug, memantine, and also discovered and cloned the factor MFC2, whose insufficient mutation for nitrosylation contributes to autism, Alzheimer's, and Parkinson's disease. So all his work has been kind of covering different diseases that we are all interested in. And he has received multiple honors and awards through his career. So we are very happy to have you here. I have the pleasure of many years ago working with Stuart, and we are very excited to hear in your new research.
Well, great, thank you. So we're going to do something a little different today. We're going to talk about, it's kind of mind-boggling actually, how a single neuron, one neuron, can predict a disease like Alzheimer's, one neuron, and also give you potential insight into new targets. So that's what I'm going to do, but I've had a lot of help to do this. So as Paula described, I'm both with you there and over here at Scripps, and I'm joined today by my bioinformatics colleague, who I'm really honored to work with, Nick Shork. Nick has been at Scripps and UCSD, among other places. Now he's the deputy director and professor at the Translational Genomics Institute, TGen, in Phoenix, Arizona. He's also an adjunct professor here at Scripps Research. And so I will, for those of you old enough to remember the Huntley-Brinkley report on NBC News, it was the highest rated actually news program ever. I guess David Brinkley was in Washington, D.C., and Chet Huntley was in New York, and they seamlessly went back and forth. No one knew how they did it, and that's what Nick and I are going to try to do today. But when I come to the bioinformatics piece, we really need Nick to describe how we do this. Okay, so the points I'm going to make today is I'm going to describe to you the first report of a single neuron patch clamp combined with deep mass spec proteomics on the same solitary excitatory Alzheimer's disease iPS-derived neuron versus an isogenic gene-corrected wild-type control neuron. And we've actually done it on about 150 neurons, but you'll see we can get single neuron data. It was published in Advanced Sciences a couple months ago. And so you don't have to take any notes. It's all out there, but I'll try to delve down to kind of, sometimes these papers are a little dense. And then I'll tell you about our bioinformatic analysis of differentially expressed proteins, or DEPs, in the AD neurons versus the isogenic control neurons. And I'll show you that with some bioinformatics that these differentially expressed proteins actually contribute to the aberrant hyperelectrical activity that we see in single neurons. And amazingly, we can relate this back to abnormal hyperelectrical activity in human AD neurons, and also seen in AD transgenic mice and cerebral organoids, which I'll show you a little data on. They all have this hyperactivity. And that activity is associated with synapse loss, because we know when certain drugs that we and others have developed, we have this new drug I won't talk about today, but it's like, I like to say it's memantine on steroids, but it has nothing to do with steroids. It has a much improved version of memantine. By blocking that hyperelectrical activity, we can actually prevent synapse loss in a variety of models, mice, organoids, etc. Okay, so, oops. And so the main point is that we can use the single cell patch proteomics technique to predict novel therapeutic targets or possibly biomarkers of the disease if we can see them peripherally too. And so, this is our first foray into that. So I'm glad to have feedback. But again, it's a little mind-boggling that you could do anything on one neuron and try to relate it to the disease. It actually took me by surprise. But you'll see many of the proteins we come up with are already known to be involved in Alzheimer's. And then we have several that nobody knew about and we think are involved. Okay, so this is kind of preaching to the indoctrinated, but I have, and you've heard this probably from other people before, but I have a little different angle on it. So, we all say it's these misfolded or aggregated proteins in the disease, right? A beta extracellularly, tau intracellular, hyperphosphorylate tangles. But the little dirty secret, and that was actually discovered by Eliezer Maslia, Paula's mentor here at UCSD and Bob Terry 30, 40 years ago. It's unbelievable. People ignore this. And he's not the only one. Is these proteins don't correlate to how demented you get. What correlates is the degree of synaptic loss on a postmortem exam. So you have serial neuropsych exams, do a postmortem exam, it's the number of synapses that really are the most tightly. And I don't know of anything else that is as tight with how you decline cognitively. So I think we should be looking for drugs that preserve synapses. And to do that, you need to know the proteins and synapses and neurons, et cetera. Okay, so why is this? Well, I think we know this is a picture we all have from the web. But just to strike home, the 40 trillion synapses we as humans have really uniquely make us humans, right? Mediating our memory, language, emotion, the essence of being human, your ability to listen to me, to argue with me. It's everything. And so it's pretty clear we need to protect neurons. And this is what's, as I said, most closely correlates with dementing illnesses, particularly Alzheimer's. This is what I would define as disease modifying. That's a word that's bandied about around a lot. And I don't think we're there yet because I don't think we have anything that really prevents or even regrows synapses. So let's talk about how you might approach this. And so, many of us are doing this. Jerome Mertens at your place, Rusty and his group at the Salk, many of us are doing this. And all over the world. So we make these iPS cells from patient biopsies. If it's a familial case of Alzheimer's, we can correct it and have what we call an isogenic gene-corrected control, which is very powerful because you might have a point mutation. And every one of the other 23,000 genes is absolutely normal. So we have the same background. We can do this in multiple backgrounds. And then we can grow them in a 2D plate, or we can grow multiple different cell types, as I'll show you in a minute, make a mini brain or a cerebral organoid. It's not a perfect model. I mean, you could argue that, gee, an organoid is 3 or 6 months in a dish, and we're 65 years old when we get, or 55 when you get the familial forms. But the stress of being in a dish actually makes them age more quickly. So we do get certain phenotypes that do resemble the disease, including this synapse loss I talked about. But there's some other wrinkle we have that I'll tell you about. So we've been doing this for Alzheimer's, for Lewy body dementia, for autism spectrum disorder. I'll talk about the Alzheimer findings today. Many of these have been published. And, they look like little brains. They have these rosettes, which I won't tell you all the markers, but they resemble the inside-out migration of neurons in the brain. They have little things that even look like ventricles. But more importantly than markers, we can add microglia and have them crawl in. So we have the full panoply of cells. We do single cell RNA-seq analysis, and we have, obviously we don't have in this particular model the endothelial cells and blood vessels, but we have all the other cell types. I don't show the microglia here, but we've grown them in. And then one approach to this, and other people are doing this too, we can analyze the differentially expressed genes in an AD versus an isogenic control and see how that develops over time. We can even do drug discovery with something called CMAP Link 1000, which is a neural network that allows you to look for drugs that correct the abnormal transcriptomics. But transcriptomics are kind of a distance from where we are, right? They encode a protein. It would be really nice if you really knew the proteins. Not all mRNAs are even translated. So, we realized you needed to do better. And so with Nick, who's on the call, and John Yates, who I'll talk about in a minute, we said, let's see if we can do deep proteomics and see if we can do it on each of these cell types. So we started out with the excitatory neurons, and I'll tell you why we did that in the next slide, if I can get it to advance, is that when you look, so we can patch clamp in organoids in 2D cultures, but another thing we do, and Alison Murtry here at UCSD does, and others do, is we can look with a multi-electrode array at the AD versus wild type, and you see something immediately. So in a wild type, this is now an organoid in a dish that's been growing, say, 3 months. And here's the heat map. Here's the actual electrical activity, the spikes. There's 64 electrodes, 1 to 64. This is time, and you can see, and each electrode is hitting an individual organoid. And you can see there's some activity, there's some spikes, but you can immediately see that the familial cases, PS1 in this case, APP Swedish, have much more electrical activity. They actually have bursting. And it's funny, Howard, I'd be curious if you were also taught this. When I was taught neurology in Boston at, I think, a pretty good place, I was told that Alzheimer's patients didn't have seizures, even subclinical seizures. And it's wrong. I mean, Leonard Mookie, I think at UCSF, Gladstone deserves a lot of credit for this. He first noticed it in his, it was actually made here at Scripps, in his J15 APP mouse model. And people said, oh, it's a bad model. And then they started doing careful EEGs on a lot of AD patients. And they have hyperactivity. A big majority of them show this hyperactivity, and some have subclinical seizures. And so, interestingly, we can reproduce this in the organoids, and even in a 2D culture. And so we have some semblance of looking at a functional readout. We're not in a vacuum in a dish, which is often a danger when you make a reductionist model. So in the organoid, we see this hyperactivity. And then we said, can we do it even in a more reductionist way? What if you record from a single excitatory neuron? And so we started this new technique that I'm going to tell you about now. And here's the workflow for the technique. So we're now looking, we're going to record with patch clamping from a single excitatory neuron, an AD neuron, isogenic wild type neuron, record from about 150 of them, and then run proteomics on them. I'll tell you about it. That's done by John Yates here at Scripps, who's a fantastic max spec guy. Would be with you today, but he's giving a talk somewhere else. And here's the workflow. So we patch clamp the individual neuron. We can measure excitatory postsynaptic currents and also spikes. Here's a sample recording here. And if you look at wild type versus the red is the AD neuron. This happens to be PS1. They have a lot more activity. You can just take the area under the curve and you can see there's a lot more activity, just like you saw in the entire organoid. So it's a little microcosm. And then we pick the neuron up and we suck it back into the pipette. We expel it into a microtube with trypsin, and we perform LC Orbitrap mass spec. And we can see a couple other people have tried this, but nobody's done it like this. So we can see almost 2,500 proteins in a single neuron, in one neuron. Okay. And so not every protein is expressed in every neuron. We have some QC, quality control. You have to have a certain number of proteins for us to count you, et cetera. And that you can read about it in that paper I told you about. So I wanted to share with you today some of the data we get out of it, because it's kind of surprising. And I think it's really encouraging that you could unbelievably model a disease with one neuron. Okay, so here's the initial kind of data we see. And so there's a Venn diagram with our wild type neurons or AD excitatory neurons, and shows the number of proteins. And you can see there's an overlap, but there's also some distinct ones. And as I said, the most we see is about, I think, 22,251 proteins have been annotated in this way. We actually see enough of the protein to analyze about 1,400 or 1,500 proteins that we really have confidence of, that we can see repeatedly in multiple neurons, single neurons at a time. And so this is how many we see. This is the number of neurons recorded and the number of proteins. And so on average, we see just short of 500 proteins, but on average in any given neuron. So I got to move the pictures of you guys because you're in my way. And so now if you just plot out on this volcano plot, the proteins that are upregulated in an AD neuron versus wild type are downregulated, you get some obvious results that kind of gave us confidence the technique might be interesting. So ApoE is high, and certain growth factors like BDNF are low. Well, that we know that in the disease, so that's kind of cool. And then we find some other things that have been associated with Alzheimer's disease before. So clustering. So that functions as an extracellular chaperone. It's known to contribute to lipid transport, which have become very important recently because of ApoE in Alzheimer's disease. It also is important in immune modulation. It's been previously implicated in AD pathogenesis because it affects amyloid aggregation and clearance. And so that's a high hit. CALR is a calcium binding protein. We know calcium is important. There's a whole set of metabolic proteins. I'm not going to go into each one, But we and others have recently published that metabolism is not normal in Alzheimer's, Lewy body dementia, HIV dementia. One of the principal reasons for this, you actually mentioned it, Paula, is they're aberrantly S-nitrosylated TCA enzymes because of the hyperelectrical activity. It turns on NOS, nitric oxide synthase. And so we kind of thought that there would be you turn off an enzyme, there's a normal feedback to make more of it. So you are making many more of these TCA enzymes and related enzymes. And so that kind of fit things already knew. But then we said, maybe we can do, with Nick Shork's input, a detailed bioinformatic analysis and really milk this more. And so what I'm going to do now is, before I tell you about linear regression, I'm going to have Nick kind of describe the technique. So Nick, you want to tell a little bit about why we used linear regression analysis?
Yeah. Pretty standard here. We'll get a little more complicated later when we do the mediation stuff. But this essentially was fitting what amounted to a linear regression model, as Stuart said, where the predictor variable was mutation status and the dependent variable was the protein abundance. And we could see that some proteins, as Stuart had argued earlier, were overexpressed and some were underexpressed. Then what we did, which many people are familiar with too, if you're at UCSD and you interact with Trey Eidecker, he does a lot of this. If you're at Scripps, then Andrew Su is probably someone you'd interact with a lot. You can take the proteins that are most differentially expressed and just ask if they are contributing to, say, one process or a pathway as a group. So that pathway is enriched. That's the term among the most significant proteins. And that little kind of clustergram to the right kind of reflects some of the relationships between the proteins that we had identified using StringDB and a few of the other databases there. So to Stuart's point, and without belaboring the issue, we found things that made sense biologically, as opposed to just having a bunch of one-offs that might be artifacts or false positives. That's the bottom line.
Let me drill down on a few examples, and you'll see where Nick is going with that. And so we found some expected things, but then we found some novel things that I think are very important because they give us potential novel drug targets. So let's just drill down on a few of these. So now we're using this AD mutation, right? We're looking at the protein expression with regard to if you have an AD mutation or you're normal. So in the AD mutation, we saw an upregulation of these proteins. So let's just talk about a few of them. This ASAH1 or N-acylsphingosine amidohydrolase. Wow, a mouthful. One is, that's a really important enzyme you may or may not know about. It's very important in making senescent cells, which is a really hot topic now in cell loss and neurodegenerative diseases. In this particular case, it functions as an acid ceramidase. We know that ceramide is toxic to neurons, and it breaks down ceramide. It's actually, the level of ceramide itself has actually been linked to apoptosis and other forms of cell death in Alzheimer's disease. So we realized that that is an interesting potential target because since it involves a sphingolipid and ceramide metabolism. EZR1 is important in cell adhesion. It's very important. It's known in cancer, and it wasn't known that it was elevated at all in Alzheimer's disease. So there's a novel one. We have to figure out what it does. Let's look at ApoE is on that list again, so it gives us some confidence that this is right. GFAP, you might say, how can that be in a neuron? It turns out there's a big proponent of people that think, particularly with the PS1 mutation, that you never differentiate properly. And you're in some kind of primordial neural stem cell state. And we know that neural stem cells express GFAP. So it's not just astrocytes. So that one actually makes some sense. People have been looking for evidence for that. I think that's some independent evidence. Let's look what's downregulated. CBX5 is a chromobox protein homolog. It represents a gene silencer. That's already been implicated because its disruption has been known to be associated with loss of tau function. So that was one that was already known in Alzheimer's, but people had seen it, the message, but nobody knew the protein was actually down as predicted. BCLF1 is a transcription factor related to BCL2, again, protective protein. It's down, so it kind of makes sense. And these, might actually represent drug targets if you could turn them back on again, right? So that's the kind of information we get from this. There's a full list in the paper of many, other proteins. And this shows the stringency bar 0.05. Above that would be significant. Okay. Yeah, so that's kind of, and then as Nick was saying, maybe I can blow this up here. Yeah, you can see the string analysis tells you the relationship of these. Oh, there's another one to talk about. Howard, you'll be entertained by this. So we all learn in neurology hexosaminidase A. A and B. A is the one, Tay-Sachs disease. Okay, we all know this. We all know it. Hexosaminidase B goes up in Tay-Sachs disease. Nobody knows why. And it's been implicated in other neurodegenerative diseases. Nobody knows why. And indeed, we found it up, and that was not known in Alzheimer's disease, but it was one of the highest overexpressed proteins in the AD mutation. So that's kind of cool. Okay, so we talked about this linear regression model. Now we're going to do the linear regression model a little bit different. Remember, before we looked at the dependence of the protein on the AD mutation. Now let's look at the dependence of it. We know that the proteins are changed in AD, so we're going to look at it versus protein abundance. And we're going to look at that versus this hyperelectrical activity I told you about. Is there any correlation with the hyperelectrical activity and the protein abundance in the AD mutation? Got it? And when we do that, we get some really interesting answers that, again, had been implicated in AD, but we didn't know it was somehow related to the hyperelectrical activity. I kind of hinted to you it was before because of the S-nitrosylation work. But so this is really fascinating. So the major proteins that are up are all redox active proteins. And what they do is they try to fight aberrant redox reactions like S-nitrosylation, sulfhydration, sulfonation. All these reactions we and others have now published occur in Alzheimer's and other dementing illnesses. So for example, in nitrosylation, you're sticking NO onto protein on a sulfur, on a cysteine, so you get an SNO, sulfur, nitrogen, oxygen. We call these snow proteins. My joke is that when I moved from Boston to La Jolla, I thought it would never snow again, and I was wrong. It's snowing. There's 10,000 of these proteins, and there's about 1,500 aberrantly expressed in Alzheimer's disease. And so this is the way you fight them. Obviously, you're not fighting them well enough because you get the disease. But PRDX1, that's peroxyredoxin. There's some others. Thyroredoxin is down in here somewhere. There it is over here. So these are upregulated. Other things that are upregulated also make sense in terms of these S10A7 and S10A9, or you may know them as S100A7, A9. There's also S100A10 and 11. These are all related proteins that are involved in inflammation. They're made in neurons, but they turn on, they get released and make inflammatory pathways in microglia. They're also S-nitrosylated to activate them, something we're studying. Okay. Let's look at a few others. Cathepsin D, we know from Randy Nixon and other people's work, this is very important in autophagy and lysosomal pathways. And so a lot of these make sense. And then if you go over to the proteins that are downregulated with regard to this hyperelectrical, you also get some interesting ones. You get histone proteins, things that control histone proteins, which are known to be important. This GPM6A is a glycoprotein that's on the neuronal surface, and it's thought to facilitate calcium channel activity and synapse formation or synapse destruction. And it had been previously, GWAS studies had previously linked it to Alzheimer's disease, but nobody knew its protein levels were actually decreased, which would make sense. And so a lot of them make sense, and they might be novel targets again. So again, I can blow up these pathways, but I had so much trouble with Zoom, I won't do it. But it also shows other proteins that are linked here, and might be targets. If you can't target that protein, you can target a related protein. Okay, so then the last approach I want to talk about, and we really need Nick for this, but it's the most interesting, is we wanted to ask, you have this hyperelectrical activity, and you have an AD mutation in excitatory neurons driving changes in protein abundance, either up or down. Who's driving who? Can we get at causality? Is it the electrical activity? And we know electrical activity can do this in some cases, normal cases. Drive protein, or is it the aberrant protein that's driving the hyperelectrical activity, which then leads to synaptic loss? So there's if you follow that, causality is very important there because it tells you where in the pathway you may want to initiate treatment, okay? So we turn to Nick for something called the linear regression mediation analysis, which I don't understand. So Nick, please explain it to us.
Well, in theory, it's actually fairly simple. Doing it in practice, a little complicated, but essentially, it's just as Stuart said, we wanted to get at the causal directions. So the mutation, does it perturb protein abundance? And then by virtue of those proteins linked to the electrophysiology and the currents and whatnot, then you have this aberrant currents. Or is it the opposite? Just like Stuart said, maybe the mutations induced through some mechanism these EPHIS aberrancies, and then those are accompanied by changes in the protein. So the changes are more secondary effects. For those of you that study cancer, this happens all the time. You look for differentially expressed genes in people with and without tumors, and you don't know which are actually causing the tumor and which are differentially expressed because there's a tumor in the body, right? So we could fit regression models, a series of regression models, kind of ruling out which directions is this causal chain kind of embedded with. And you see on the left panel there, this is assuming that it goes mutation, protein abundance, EPHIS, and then in the right panel, it's mutation, EPHIS, proteins, and you can see that the evidence is much more consistent with the proteins being disrupted by the mutations and that leading to the aberrant kind of EPHIS phenotypes.
So the way we
So
read
it
out
makes
here is
sense.
the red line is the line for approaching statistical significance. This is really an exploratory study. So we kind of lowered our aims on how high it had to be statistically relevant. But you can see even if you lower it, there's no proteins whereby the proteins are driving the hyperelectrical activity. But there's some interesting proteins that seem to be linked where the proteins are driving the electrical activity as opposed to the other way around, the electrical activity driving the proteins. So here's the protein abundance in AD driving the electrical activity. And we see proteins that we've already talked about, which gives us confidence that this analysis has some merit. So again, these redox proteins, thyrodoxin, we see this ASASH1, which remember, that's a sphingolipid ceramide protein. We talked a little bit about histone proteins. There it is. And maybe most importantly, we see growth factor proteins are downregulated. We know that's true in the disease. That was the basis of Mark Dujinski's gene therapy proposals, right? Here's a really interesting one that only in the last few years we know is important. TPTRFE. That's the major thyrodoxin binding protein. It's down. So iron is up. And we know that ferroptosis is a major pathway to cell injury and death in Alzheimer's disease. And this is probably one of the reasons. The binding protein that's protecting you from free Fe3 plus is depleted. That would be a fantastic target. We've actually already thought of that target for other reasons, okay? So what we're trying to do here is use this to kind of predict targets, see if they make sense with the disease. And now there's another implication here, and this is Howard, why I'm going to call you and talk to you somewhere, is we do have developed drugs that block the hyperelectrical activity. And if it's the protein abundance that's driving the hyperelectrical activity, that's then driving the synapse loss, which is all of our data, if we just treat the hyperelectrical activity, we may do a lot of good. We may be able to protect synapses. So this study made sense in terms of that. Okay, so we have lists of proteins. You're welcome to go to that paper in Advanced Science and see them, but I wanted to kind of hit the high points. I hate it when people give talks with thousands of volcano plots and tables with 4,000 proteins. You can just go look at them, but I wanted to hit the high points to kind of show you the idea of the analysis and to show you some of the proteins that have come out, and have fashioned our thinking about how we might want to, in the future, approach the disease in a disease-modifying way that protects synapses. Okay, so what's that? Okay, so let me tell you what I've told you, and then we can, I particularly wanted to have a lot of time to talk about this because, any new technique should be contentious, and we want to talk about that. So we've tried to show that single excitatory iPS-derived AD neurons, in this case from familial cases, compared to their wild-type isogenic counterparts, so hyperexcitability. That's interesting because you see that also on human patient EEGs. We see that a single iPS-derived AD neuron, we can patch clamp them. We can, by MS proteomics, get more than 2,250 proteins in a single neuron, and then kind of mind-boggling. You can do a linear regression analysis of a single neuron and show pathways that we think are implicated in the disease, and even come up with some targets, some new targets, some old targets that might affect the disease. And these targets are related to redox chemistry, metabolism, inflammation, and cell death pathways such as ferroptosis. We gave you examples of each of the proteins we found. And then this mediation analysis, which is a little more dodgy, but I think really exciting, suggests that the differentially expressed proteins due to the AD mutation are driving the hyperelectrical phenotype. And we already have, we and others have published evidence that hyperelectrical phenotype is a major component of synapse loss. So this implicates if we can treat this hyperelectrical phenotype, and I think we already can, that's going to be protective to synapses. So collectively, we suggest some pathways and specific targets that may have therapeutic potential, some of which were known, but a big number of which weren't known. Now we can do this for the other types. We can do it on inhibitory neurons. We can do it on other cell types. So we're just starting to have fun with this. Yeah, so that's kind of where we are. This is where it's done. Come over and visit. We're actually moving to that new building on the corner of Genesee and North Torrey Pines, the one that hangs out on the road that you almost hit. Somebody's going to hit us. Somebody's going to go right through that light into my lab. And I'm on the first floor right at the light. So I already said this is going to happen. Anyway, we've had a lot of help with this, but a lot of people, I mentioned Nick prominently and John Yates, but a lot of people have contributed to this. So I'm glad to discuss this. I hope there's some soothsayers that don't believe anything we said. Nick, you'll defend us, right? And then, maybe other people have other ideas where this kind of technique can go. So thanks for listening.